welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 29th June 2021. As we are discussing about chapter 2 biological classification, in this session we will be quickly go through what we have talked, what we discussed yesterday, and then go to the new concept. As we said yesterday, biological classification is the scientific procedure of arranging organisms into groups on the basis of their similarities and dissimilarities, and placing the group in a taxonomical hierarchy of categories. This is what we said. And for this biological classification, we must be thankful to many great scientists. So to begin with, Aristotle's classification. Aristotle was the earliest to attempt a more scientific basis of classification of organisms. He classified plants to trees, shrubs, and herbs, and animals into two groups, those with red blood and animals without red blood. So you got animals with red blood and animals without red blood. So animals with red blood, naturally all of us, mammals, lizards, birds, fishes will come, and animals without red blood, but they are classified into two, hard-bodied and soft-bodied. So you got hard-bodied, that is insects, soft-bodied, shell, and no shell. If there is a shell, it is shellfish. If there is no shell, jellyfish. So this is how we classified. But it has got its own demerits. It is not accepted by majority. So there was a proposal by another scientist by name Carl Linnaeus, and he proposed two kingdom classification, and he broadly made all organisms into plants and animals. So kingdom Plantae and kingdom Animalia. But that also has got defects. What are the defects? As we discussed yesterday, what about fungi? Where to place fungi? And what about bacteria? So there was many drawbacks in the two kingdom classification. Like we say, prokaryotes, that is bacteria, cyanobacteria, and eukaryotes. I said karyon means nucleus. Prokaryotes means organisms that are having not very clear nucleus. It's something like nucleoid is present without a proper nuclear membrane. Whereas eukaryotes, when we say eu means clear nucleus is there, eukaryotes. So fungi, mosses, ferns, gymnosperms, and angiosperms, these are having clear-cut nucleus. So these were included under plants based on the presence of cell wall, but they are widely differed in other features. Second, it included unicellular as well as multicellular organisms in the same group, like Chlamydomonas and Spirogyra were placed under algae. It did not differentiate between heterotrophic fungi and autotrophic green plants. Everything is kept under Plantae, kingdom Plantae. Fungi have chitinous cell wall. There is a chemical called chitin in the cell wall, while the green plants have cellulozoic cell wall. Cellulose is present. So these are the defects in the two kingdom classification. So there is a need for further. It was three kingdom, then it came four kingdom, and finally the most accepted version, as I said yesterday, is five kingdom classification. This is proposed by R.H. Whittaker in the year 1969. It includes Monera, Protista, fungi, Plantae, and Animalia. So this classification is based on, as we discussed yesterday, cell structure, thallus, organization, mode of nutrition, reproduction, and phylogenetic relationship. Phylum means evolutionary relationship. So basically, Whittaker has classified all the organisms into five kingdom. Kingdom Monera, kingdom Protista, kingdom fungi, kingdom Plantae, and kingdom Animalia. And how he has classified this? Based on 5 characters: cell wall, nuclear membrane, body organization, and mode of nutrition. So under Monera, cell type prokaryotic, protista, fungi, Plantae, Animalia all are eukaryotic. Means nucleus is very clear. Second character, cell wall. In Monera it is non-cellulosic. Cellulose is not there, but polysaccharide plus amino acids are present. In protista it is some, means it is a mix. Whereas in fungi it is chitin. Just now we said chitinous cell wall. Whereas in plants it is cellulose. In our case it is absent. In animals there is no cellulose. Nuclear membrane, since it is prokaryotic, naturally there is no nuclear membrane, absent. Rest all present. Body organization, they are all basically single cell, unicellular organ, so it is cellular. And protist also cellular. But others, it is multicellular, fungi. In the case of plants, Plantae, tissue and organs have come. In our case, not only tissues and organs, even organ systems like digestive system, respiratory system, circulatory system has come. Mode of nutrition, it is autotrophic, means they prepare their own food because of presence of photosynthetic substances. So it can be through photosynthetic, chemosynthetic, as well as saprophytic, parasitic, all types of nutrition will be there. Whereas in protista, it is basically autotrophic. In fungi, of course, it is fully heterotrophic. Fungi cannot prepare their own food because they don't have chlorophyll. So they have to depend on others, either saprophytic or parasitic. Being parasites, they depend on other organisms for their food. And also they are saprophytic. They eat dead and decay matter and obtain energy. Plantae, all are autotrophic, photosynthetic. In our case, Animalia, it's holozoic as well as saprophytic. And the first kingdom we discuss, kingdom Monera. This is you can remember this as bacteria family. Kingdom Monera is bacteria. Monerians means bacteria. Bacteria are the most abundant microbes, as all of us know. They are present everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans. And many are parasites. They are present in our own body, in the intestine, in the stomach. So they are present everywhere. So basically, kingdom Monera, which is a bacteria family, has got bacteria with 4 types of shapes. So based on the shape of the structure, shape of the cell, bacteria is classified into 4 types. Round or spherical ones, they are called coccus, round structure. Rod shaped structure bacteria, they are called bacillus. Comma shaped one are called vibrium or vibrium. Spring like structure, if the bacteria's body is like a spring, then we say spirillum. So you have got various types of bacteria. You must have heard some of the names like Streptococcus pneumoniae, which causes pneumonia, lung disease, Clostridium tetanus, Trypanosoma palladium, Vibrio cholerae, that causes cholera, Salmonella typhi. So all these you must have heard. Clostridium, Staphylococcus, all these names of the bacteria, they are very familiar with us. Basically, what is the structure of a bacterial cell? So outside, basically you have got a cell wall, a capsule will be there, and then plasma membrane. See, 3 layers are there. A capsule is there, cell wall is there, plasma membrane is there. And inside there is a cytoplasm. In the cytoplasm, you've got ribosomes, which are helping in protein synthesis. Plasmids are there, and villi, locomotor structures are there. And a very whip-like big structure will be there, that's called flagellum. And since they are prokaryotes, where nucleus is not very clear, nucleus-like thing is there. That nucleus-like thing is called nucleoid. So it is basically a circular DNA. So bacterial structure is very simple. but they are complex in behavior and show metabolic diversity. Some bacteria are autotrophic, they synthesize food from inorganic substances. Majority are heterotrophs, they depend on other organisms. They don't synthesize their own food but depend on other organisms or dead organic matter for food. So basically kingdom Monera is classified into two major groups. That is, one group is archaebacteria. Archaebacteria, it means ancient bacteria, old bacteria. And second one is eubacteria, that's modern one. So archaebacteria is further classified into three types: halophiles, thermoacidophiles, methanogens. This is based on their habitat. Eubacteria, on the other hand, is classified based on the nutrition, autotrophs and heterotrophs. In the autotrophs, it is photosynthetic autotroph, chemosynthetic autotroph. And heterotrophs, there are two, parasitic and saprophytic. Now moving ahead, archaebacteria, they live in harshest habitats such as extremely salty areas. So if the bacteria are living in salty sea, then we say halophiles. So the halophiles are salt-loving bacteria. They are salt-loving bacteria as they are found to live in environments with high salt concentration. And the second one, hot springs, thermoacidophiles. So thermoacidophiles live in extremely acidic environments where pH is less than 2 and that have extremely high temperatures up to 110 degrees centigrade. See, look at that. And they are found in hot sulfur springs. On the other hand, methanogens are obligate anaerobics. So in biogas fermenters, these bacteria produce methane. So methanobacterium, methanococcus, these are the examples that we can quote for methanogens. So archaebacteria have a different cell wall structure for their survival in extreme conditions. Just imagine surviving at 110 degrees centigrade. So they need to have special cell wall. Methanogens are present in the guts of ruminant animals like cows and buffaloes. They produce methane, biogas, from the dung of these animals. You can see the picture. You can see the presence of methanogens in the cow and buffaloes. Continuing, eubacteria. Under the kingdom Monera, second type is eubacteria. So in this eubacteria, these are true bacteria having very rigid cell wall with a flagellum. Flagellum helps in locomotion, motor. They include autotrophs, that is photosynthetic and chemosynthetic, and heterotrophs. So in photosynthetic autotrophs, example we can say cyanobacteria. They have chlorophyll similar to that of green plants. Like you can see here the examples also given: Anabina, Oscillatoria, nostoc. So basically these are called cyanobacteria, or common name is blue-green algae. So it is basically unicellular, colonial or filamentous, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Algal bloom we say. Some of them fix atmospheric nitrogen in specialized cells called heterocysts. This is a one more question. What are heterocysts? So these have specialized cells. Heterocysts are specialized cells. They help in fixation of nitrogen in the atmosphere. That's how nostoc and Anabina. The farmers can use this. It can be very, very useful. And continuing the second type under eubacteria, chemosynthetic autotrophs. So you can see green sulfur bacteria, purple sulfur bacteria, and you can see the perfect example of Vinivibrio stagnis pimentis. They oxidize inorganic substances such as nitrates, nitrites and ammonia and use the energy that is released for ATP production. Since they are using the inorganic substances, chemicals, we say chemosynthetic autotrophs. So basically they break these inorganic substances. When they break that, whatever energy that is released, that released energy will be used for ATP production. So with that ATP they survive. So they help in recycling nutrients like nitrogen, phosphorus, iron, sulfur. That's why we are using the name green sulfur bacteria, purple sulfur bacteria, like that. Continuing eubacteria, the heterotrophic bacteria, they are most abundant in nature. The majority are important decomposers. So basically the heterotrophic bacteria help in decomposition. Hetero means different. They depend on different bacteria. Continuing with that, heterotrophic bacteria, how do they impact on us? What are the uses of this? So basically they are used to make curds from milk. So the bacteria help in our day-to-day life in converting the milk into curds, and then they are helpful in production of antibiotics. Whatever antibiotics that the medical community is using, they're all basically from the bacteria. Fixing nitrogen in legume roots, leguminaceae roots, and some are pathogens. Of course, they cause diseases like cholera, typhoid, tetanus, and citrus canker in citrus fruits. All these are there. That means we understand that they are helpful as well as dangerous. Like rhizobium, very very helpful. Salmonella is dangerous. Vibrio cholerae is dangerous. That means eubacteria are bacteria that are useful as well as harmful. And further, how do they reproduce? Basically, bacteria reproduce mainly by fission. Fission means breaking of the body. And I said, in fission there are two things that take place. One is the nuclear division, karyokinesis, followed by cytokinesis. So in fission, compulsorily these two processes will take place. So basically the fission is the one where the bacteria will divide into two parts and each part becomes a adult bacteria. Under favorable conditions they produce spores. You can see here the spore formation, and these spores under favorable conditions will generate into a new adult. But in case the conditions are not favorable, I told you, remember, when going gets tough, the tough gets going. The spore becomes a cyst, and the cyst will survive as long as unfavorable conditions are there. Once the favorable conditions return, the spore will break up and release thousands of spores. That is the thing. So they also reproduce by a sort of sexual reproduction, that is a DNA transfer from one bacterium to other. So we have got another important group of bacteria in this. They are called Mycoplasmas. These are organisms without a cell wall. There is no cell wall in Mycoplasma. They are the smallest living cells known. So this is a normal question, one more question. These are the smallest living cells known, Mycoplasmas. They can survive without oxygen, and many are pathogenic in animals as well as plants. So Mycoplasmas are the simplest and the smallest of all the free living prokaryotes. They can survive without oxygen. Due to the absence of cell wall, the organisms can keep on changing their shape and are pleomorphic. Morphism, morphology, pleomorphic. They keep on changing the shape. The organisms are often called MLOs, means mycoplasma-like organisms, or they have got another name, PPLOs, that is pleuropneumonia-like organisms. PPLO, I will repeat, pleuropneumonia-like organisms. They form characteristic colony with fried egg appearance. Okay, that is the characteristics of this. And of course, we have got actinomycetes in this. Actinomycetes are mycelial, means aseptate, branched, filamentous bacteria, which form radiating colonies in culture. Hence, they are called ray fungi. Example, streptomyces. produces antibiotics such as streptomycin, chlorophenicol, tetracycline, all this. So Streptomyces is the largest antibiotic producing genus. So these are the things you should know under kingdom